Hello student and welcome back to Top Scholars. In today's session, we are going to discuss the PYQs from basic biology that were asked between the year 2023 to 2025. Alright children, so let's start with the topic cell, the structural and the functional unit of life. Let's look at the weightage of this topic children. So as we can see over here, in the year 2023, 1 marker question was asked and 2-marker question was also asked, okay, and in 2024 only 1 question of 2-marker was asked from this topic. So now we'll be beginning with the topic, the structure of the chromosomes, cell cycle and cell division. So let's quickly look at its weightage. So as we can see over here, in year 2023, 2 questions of 1 mark were asked and 2 questions of 2 marks were asked. In year 2024, 2 questions of 1 mark and 1 question of 3 mark was asked. And in year 2025, we had 4 questions of 1 mark each, 1 question of 2 mark and 1 question of 3 marks. Alright, so the highest weightage from this topic was asked in the year 2025. Okay, now let's move on. Now we will start with the topic genetics, the basic fundamentals. But before looking at the questions, let's look at the weightage of the topic. In year 2023 children, 4 questions of 1 mark were asked and 2 questions of 2 marks were asked. In year 2024, 2 questions of 1 mark and 1 question of 3 mark was asked. And in year 2025, 3 questions of 1 mark and 1 question of 3 marks was asked. Now let's look at this question. The organelle that forms ester during the cell division is known as centrosome. So children, centrosome is a membrane-less cell organelle which is present near the nucleus, and it consists of 2 perpendicular cylinder-like structures which are known as the centrioles. During cell division, the centrosome, it migrates to the poles of the cell and it starts producing the aster rays, which gives rise to the spindle fibers which will help in arranging the chromosomes at the equatorial plane. Okay, now let's move on to the next question. Now, the second question is again of a very short answer category and it is of 2 marks. So, let's look at the question. The question is, what is a semi-permeable membrane? And the second question is, name the semi-permeable membrane which is present specifically in the plant cell. So, let's solve the first question. Okay children, so what is a semi-permeable membrane? Children, semi-permeable membrane is a membrane that only allows few substances to pass through it and enter into another medium. So let's look at this diagram children, for example. Okay, now this is a container. Now I am dividing this container into 2 parts, that is part A and part B by this membrane. Now, this is not a freely permeable membrane, this is a semi-permeable membrane. Children, semi-permeable means that only certain substances are allowed to move from one medium to another. And that is along its concentration gradient, that is from high concentration to low concentration. Let's say I have 5 grams of sugar in the container A. And I only have 1 gram of sugar in the container B. Now, this membrane allows the sugar to move from container A to B or B to A. However, children, here in this case, the glucose will move from container A to B. That is from its high concentration to low concentration. So, children, what is a semi-permeable membrane? Semi-permeable membrane is a membrane that allows movement of certain substances through diffusion, that is from its high concentration to low concentration. Okay children, I hope this is clear. Now let's move to the second question. The second question states, name the semi-permeable membrane which is found in the plant cell. Okay, so let's quickly look at the diagram of a plant cell. Children, when we look at a plant cell, the outermost membrane of the plant cell or the outermost covering of the plant cell, which is very strong, thick and rigid, that is the cell wall, right? Okay, please remember children, cell wall is a freely permeable membrane. Okay, now inside this cell wall children, we have another membrane, and this is the cell membrane of the plant cell. Cell membrane is also known as the plasma membrane. Do you know its another name children? Yes, you are correct. It is also known as the plasma lemma. Okay children, so the cell membrane or the plasma membrane or the plasma lemma, children, all these three are the same structure and it is semi-permeable. Okay, so what is the correct answer for the next question? That is, the semi-permeable membrane in the plant cell is the cell membrane. I hope this is clear children. So let's quickly look into our diagram again children one more time. So the cell wall is the outermost covering of a plant cell, right? It is a freely permeable membrane. That means it allows everything to come from outer side of the cell into the cell. However, the plasma membrane children, it is very specific. It will only allow certain substances to come from outside the cell into the cell. I hope this is clear. Okay. Alright. So let's move on to the next question. Now in the year 2024, let's look at the MCQ that was asked, which is for 1 mark. Select the correct answer to the questions from the given options. Duplicated chromosomes are joined at a point termed as option A is centrosome, option B is centromere, option C is centriole and option D is chromatid. Isn't this a super confusing question? All these words literally sound the same, but they are different from each other. So let's solve this MCQ using the elimination method. In elimination method children, what do? We eliminate the obviously wrong answers and with that we will come to the right answer. Okay, so let's start with the option A. Option A says centrosome. Children, now what is a centrosome? Centrosome is formed when two centrioles, which are these perpendicular structures found in a cell, they come together and they lie perpendicular to each other. This structure together is known as the centrosome. Okay, and this individual perpendicular structure, they are known as the centriole. So obviously these are the incorrect answer. So option A and option C are incorrect. Now coming to option D. Children, what is a chromatid? A chromatid is the arm of a chromosome. So when we look at a chromosome, we see this ribbon like structure. So these are the chromatids. Alright children, so here also this is not a relevant answer to our question. What is the question children? The duplicated chromosomes are joined at a point. So let's draw a complete chromosome. So this is the complete chromosome and this is its duplicated part which is formed during cell division. So the point where both of them are connected or joined with each other, this point children is our centromere. Therefore, the correct answer is option B, that is the centromere. I hope this is clear. So let's quickly revise this whole thing. The question asked over here was that the duplicated chromosomes, they are joined at which point? right? The option given over here is centrosome, centromere, centriole and chromatid. So let's quickly look at the option. What is a chromatid, children? Chromatid is the arms of the chromosomes, right? Next option is a centriole. Centrioles are this perpendicular bar-like structures and together it is known as the centrosome. So these are the obvious incorrect answer and therefore the correct answer is a centromere. Centromere is a small constriction which is present in a chromosome where both the duplicated and the original chromosomes are connected to each other. Okay. Also children, this question can be asked in an alternate way, that what is the point where the chromatids are connected to each other? The answer will still be the same, that is the centromere. So we can see over here, these are the two chromatids, they are also connected to each other at this constriction which is the centromere. So children, I hope you have understood the question why the right answer is centromere. Okay, chalo, let's move on to the next question. Okay children, now let's look at a 2-marker question which is of the category very short answer. So the question is, why are RBCs efficient in their functions even though they lack nucleus and mitochondria? So children, first let's look at the structure of the RBC. RBC children is not like a typical cell which is spherical or cuboidal in shape. It is biconcave in shape. So you all must have seen the diagram of an RBC. It's like this dumbbell shaped, you know. This is like this. This is your typical RBC. Okay, it's biconcave in shape, meaning there is a concavity from the top and there is a concavity from the bottom. This is what your RBC looks like. And it is not only the shape that makes the RBC unique, but the second thing that makes it extremely unique and different from all the cells of the body, that is, it literally have no cell organelles. Yes, it has no nucleus, it has no mitochondria, it has no ribosomes, no endoplasmic reticulum, no Golgi complexes, no lysosomes, etc., etc., etc. It's like a classroom without any benches, without any chairs, without any table, blackboard, cupboards, literally nothing. It's an empty room. And yet RBC is still a very, very, very efficient cell. Now let's understand why. Children, when you remove everything from a cell, that will give us a lot of space in a cell, right? And that will help us to load the RBC with huge amount of hemoglobin molecules. Yes, so when we say that RBC has no cell organelle, that does not mean it's a lazy cell. No, it is going to replace it with huge number of hemoglobin molecules, children. More the hemoglobin, children, more will be the oxygen carrying capacity of a cell. That means more the hemoglobin the RBC has, more amount of oxygen it can carry to the cells. Isn't that a great thing? Children, let me tell you one thing. Whatever air we inhale, 97% of the oxygen that comes in that air is carried by the hemoglobin, that means by the RBCs. So even when RBCs don't have the cell organelles, RBC is still working very hard. It is carrying 97% of oxygen to all the cells of the body. Isn't that great? Isn't that what we require? Imagine if our cell was filled with all the cell organelles. We would have very little space for hemoglobin. So let me draw a small cell. Okay, let's say this was my normal typical RBC. If it has this nucleus, it has this DNA inside it. Along with that, it has an endoplasmic reticulum. Along with the endoplasmic reticulum, there are these Golgi complexes, then there are the ribosomes, and then there is a lysosome, and so on. So if my RBC was filled with so many cell organelles, imagine the space I would have for hemoglobin. Way less, right? But if I have this RBC with literally nothing inside, so imagine the amount of hemoglobin I can carry, a huge number. And more the hemoglobin, children, present in a cell, more will be the oxygen carrying capacity of a cell. And that is why the RBCs are completely devoid of cell organelles. Alright children, now let's look at the second advantage, that when there are no cell organelles present in a cell, the cell becomes highly flexible. Do you know that RBCs are actually called as the gymnast of the body? Yes, because they are the most flexible cells, because they are the only cell without any cell organelles. Now you might think that why do RBC need so much flexibility? What is the point? Right? So let me explain that thing to you. Children, the RBCs have to pass through the narrowest capillaries of the body. Right? These capillaries are the narrowest blood vessels. Its diameter is literally half of that of the RBC. That means RBC is 2 times more the size than that of a capillary. So do you think the RBC would be able to pass as it is without performing any stunts through the capillaries? No, right? So the RBCs needs to be flexible. In fact, you will be surprised that when RBCs have to pass through the capillaries, it literally fold upon itself like a C. So your RBC, which is like this, it will fold upon itself like a C so that it can pass smoothly through the capillaries. So RBC would look something like this when it is passing through the capillaries, and then it will flow through it. Okay, so there are 2 reasons why RBC is still a very efficient cell of the body even when it does not have nucleus and mitochondria. Reason number 1, more space to carry hemoglobin, thus more oxygen. And reason number 2, more ability of a RBC to be flexible so as it can pass smoothly through the capillaries. I hope this is clear, children. Now let's move ahead. Now we've finished all the PYQs asked from the previous topic. Now let's move to the next topic, that is the structure of chromosomes, cell cycle and cell division. A topic lots of students get confused, but I'm here to make it simple for you. So let's start with it. Children, from this topic, let's look at the weightage. It's quite high, roughly from 6 to 9 marks of questions are asked each year from this topic. Okay, so in the last 2 years, that is from 2023 to 2025, let's look at the category of questions which are asked. So in the year 2023, children, 1 marker questions were asked, total 2 in number. 2 marker questions were asked, which were also total 2 in number, and therefore the weightage is total 6 marks. Okay, in the year 2024, total 1 question was asked of 1 mark and 1 question was asked for 3 marks, and therefore the total weightage is 4 marks. Okay, and in the year 2025, they have literally topped the charts. We can see over here, total 4 questions were asked of 1 mark each, 1 question of 2 mark each, and 1 question of 3 mark. Alright, so total 9 marks of question is asked, and therefore you need to do this topic really well. So we are going to do all the PYQs that is asked in order to improve your confidence and clear your concepts. So let's start with it. Over here, we can look at the same thing, children, in the graphical format. So we can see over here, in the year 2023, 2 questions were asked for 1 mark each and 2 questions were asked of 2 marks each. In year 2024, 1 question was asked for 1 mark and 1 question was asked for 3 marks. And in year 2025, 4 questions were asked of 1 mark, 1 question was asked for 2 marks and 1 question was asked for 3 marks. Alright, so I hope this made the importance of this topic even more significant in your minds. Now, let's look at the questions. So, first we'll start from the year 2023. We are going to look at an MCQ which was asked for a 1 mark. Let's look at it. Select the correct answers to the questions from the given options. During synthesis phase of the cell cycle, synthesis phase meaning, children, S phase of the cell cycle, more of option A, RNA is synthesized. Option B, RNA and proteins are synthesized. Option C, DNA is synthesized. And option D, glucose is synthesized. If you are not able to answer this question, don't worry. Let me break this down in simplest words. Children, we know that cell division is broken down into 2 phases. One is the interphase and the second is the dividing phase. Okay, children. Interphase earlier was referred as the rest phase. Scientists thought that cells are just chilling in the interphase. but later on it was disapproved, and we found that cells are highly active. They are preparing themselves to enter into dividing phase. Okay, so interphase is a phase where lots of changes are happening in the cells. Interphase is further divided into three more phases, children. That is the G1 phase, S phase and the G2 phase. Alright. Now G1 phase is known as the first growth phase, S phase is known as the synthesis phase, and G2 phase is known as the second growth phase. Okay. Now in this phases, children, there is synthesis of lot of raw materials is happening, because when a cell has to divide and give rise to two daughter cells, you need to give everything to both the daughter cells, right? That means you need to have two DNA copies. That means you need to have two nucleus, two mitochondria, two endoplasmic reticulum. So everything needs to be duplicated, and all that duplication of the cell organelles and the raw materials required for that is seen in the interphase. Okay. In the G1 phase, children, we see lots of RNA synthesis, lots of protein synthesis, and other cell organelles also duplicate. And similarly is seen in the G2 phase, that is we see again some more RNA and protein synthesis. So activities in G1, G2 are slightly similar. However, S phase is a very unique phase where only and only, please hear my words carefully, only and only DNA is synthesized or DNA is duplicated. See children, a parent cell already has its own set of DNA, right? Now this parent is reproducing, it is giving rise to two daughter cells. That means both the daughter cells needs to be given its own DNA. That is only possible if a parent cell, it duplicates its original DNA. So let's say this is the original copy of DNA. I'll pass this through the Xerox machine and I'll get a duplicate copy. Only then I can give it to both my daughter cells, right? Therefore, in the S phase, this Xerox or this duplication or synthesis of new DNA is seen, and therefore S phase is known as the synthesis phase. Okay. So over here, what is happening? The DNA is replicated or the DNA is synthesized. Okay, the DNA is replicated or the DNA is synthesized in the S phase. Please keep one thing in mind, that obviously the quantity of DNA is becoming double, but the ploidy does not change. That means if the organism is diploid, the organism will still remain diploid. Meaning if the organism has 46 chromosomes, it will still remain 46 chromosomes. Okay children, so over here the correct answer is DNA is synthesized. Okay children, so I hope you understood this. Alright, why is option D incorrect? We have talked about why option A and B is incorrect, right? Why is option D incorrect? Children, glucose synthesis is not seen during cell division. It takes place quite before cell division, right? Glucose is synthesized. How? If it's a plant cell, then it is by breakdown of starch into glucose, right? And if it's an animal cell, then it is during the breakdown of glycogen into glucose. Okay, so this breakdown to form glucose is not seen during the synthesis phase or the S phase. Therefore, the correct answer is option C, that is DNA is synthesized. I hope this is clear. Okay, so now let's move to the next question. Now this is again asked in year 2023. It's a 2-marker question. So let's look into the question. Differentiate between karyokinesis and cytokinesis. So let's solve this question. Children, cell division. Let's again draw this quick mind map. Cell division is divided into two main phases, that is the interphase where the cells do not chill, please keep in mind, it's actively working hard, and the dividing phase. Dividing phase is also known as the mitotic phase. Okay. Now, the dividing phase is further classified into two more subphases, that is the karyokinesis and the cytokinesis, which is a question. So, let us answer it in detail now. Children, karyokinesis, this word comes from merging two words. Word number one is karyon. Karyon means nucleus, and kinesis, that is the word number two, it means division. So, karyokinesis stands for nucleus division or nuclear division. Alright. The word cytokinesis similarly comes from merging two words. One is the cytoplasm, okay, and the kinesis is again division. So, cytokinesis stands for division of cytoplasm. Alright. So, in karyokinesis, children, the nucleus is dividing to give rise to two nucleus, and in cytokinesis, the cytoplasm divides to eventually separate the two daughter cells. I will show that to you in a diagram, but let me quickly tell you all the phases of karyokinesis. In the karyokinesis, children, we have the prophase, then we have the metaphase, then we have the anaphase, and the last we have the telophase. So, once all these phases are done, children, now let me show you the diagram. Once all these phases are done, children, by the end of telophase, we get to see two nucleus inside a cell. Now, you will say, how did this exactly happen? Look at the parent cell, children. This was a parent cell who decided to reproduce, who decided to divide. This is one single nucleus of the parent cell. Now, this parent cell will first do karyokinesis. What will this parent cell do first? It will do karyokinesis. That means it will do prophase, metaphase, anaphase and telophase. And at the end of telophase, children, we can see that we are seeing two nucleus now in the parent cell, because one nucleus has divided and given rise to two nucleus. Alright. Now comes the last phase of the cell division, that is the cytokinesis. Now children, if this is an animal cell, the cytokinesis will take place in this manner, where the cell membrane will move inwards and eventually it will separate and give rise to two daughter cell having its own single nucleus. Alright children, so that will be at the end of cytokinesis. So, what's the difference between karyokinesis and cytokinesis? Karyokinesis is division of nucleus to give rise to two nucleus, and cytokinesis is division of the cytoplasm to give rise to finally two independent daughter cells. Okay. What are the steps involved in karyokinesis? We have the prophase, metaphase, anaphase and telophase. What are the different types of cytokinesis? In an animal cell, it is by the invagination method, where the cell membrane moves inwards, and in a plant cell, it is by a cell plate method, where there is a formation of a cell plate from the center and the cell plate will extend towards the periphery. So, let me also show it to you one more time. This is the invagination method in animal cell, and in a plant cell, children, we will get to see something like this. These are the two daughter nuclei which are formed. In the plant cell, there will be a formation of a cell plate in the center, which will keep on extending from center to the periphery, right? In animal cell, children, what was the case? Just drawing it quick for a quick comparison and clarification of the concept. The cell membranes were moving inward. So, it is moving in the opposite direction. So, cytokinesis in a plant cell is from the center to the periphery, and in the animal cell, it's opposite, from the periphery towards the center. Okay. So, I hope this answers your question and clarified your concept. Now, let's move to the next question. Now, this was a 1 marker question, which is a very short answer asked in the year 2023. That is, give the abbreviation or expand the abbreviation of DNA. I hope you are able to answer this already. If not, let me help you. Deoxyribonucleic acid. What is the DNA, children? DNA is the genetic material most of organisms that control the activity of each and every cell of the body. I hope, children, this is clear. Okay, let me show you a structure of single nucleotide of DNA. Okay, so DNA is formed, or a nucleotide of a DNA is formed by this deoxyribose sugar. Okay, on the first carbon we have the nitrogenous bases, and on the fifth carbon, children, we have a sixth carbon attached to it. Through that we have a phosphate group attached to it. Okay, this is what a nucleotide of DNA looks like. When I will join millions of this nucleotide together, I will get the chain of DNA. Okay, this is a deoxyribonucleotide, which is a monomer of DNA. Okay, DNA is a polymer, so you just join all these deoxyribonucleotides and you will get the entire polymer of DNA. Okay, children, I hope this is clear. Now, let's move to the next question. Now, this is again a 2-marker question asked in the same year. Let's look at the question. What is active transportation? Very simple question, children. So, children, in nature or in earth, generally, substances move from its high concentration to low concentration on its own naturally. So, let's take a container. Now, I will divide this container by using this semi-permeable membrane, and this part will become container A and this will become the container B. Okay, children. Now, let's say in the container A, there is 5 gram of glucose, and in the container B, there is only 1 gram of glucose. So, here we have 5 gram of glucose, and here we have 1 gram of glucose. So, naturally, the glucose will move from its high concentration to low concentration. That is, the glucose will move from container A into container B. This natural movement of substances from high to low concentration is known as simple diffusion. Alright, and no ATP or no energy is required because it naturally flows. Children, in the same example, if I want to reverse the movement, that is, let me draw it again here. It is the same example. There is one single container. I am dividing it into 2 using this semi-permeable membrane. Here I have 5 gram of glucose, and here I have 1 gram of glucose. Right? But now I want the movement of the glucose from B to A. Yes, I want it to move from low concentration to high concentration. That is also possible, but it needs a push because you are going opposite to the law of nature, right? If I want the movement to take place from B to A, that is from low concentration to high concentration, this is not going to happen on its own directly because this is going against the nature. Yes or no? But I can still make it happen if I push this glucose against its concentration gradient, that is from low to high. And for pushing, what will I need? I will need energy. Yes. So here I will need to spend a lot of energy to move the substance from its low concentration to high concentration. And this is exactly what active transport is about. Okay, so active transport is movement of substance across a semi-permeable membrane, low concentration to high concentration by using energy or by breakdown of ATP. I hope this is clear, children. So this option B is an example of active transport. And here this example A that we have drawn, this is the example of simple diffusion or passive transport. I hope active transport is also clear and difference between active and passive transport is also clear. Okay, students, let's move on. Now let's look at this question. Select the correct answers to the question from the given options. A male gorilla has 48 chromosomes, pay attention to the number, in each of its body cell. How many chromosomes will be present in the sperms? This is a question. So, option A is 24, option B is 48, option C is 12 and option D is 16. So, let's quickly solve this question. Children, the data given to us is that there are total 48 chromosomes in each cell of the body, right? We know that the gorilla is a diploid organism, meaning each cell in its body is a diploid cell. That is 2n is equal to 48, right? Now children, we also know that in order to produce sperm, we need to do meiosis, right? For sperm production, the cell division that is seen is meiosis. We know that meiosis is a reductional cell division. What is meiosis? It's a reductional cell division. That is, the number of chromosomes becomes half. Here the data given to us is that total 48 chromosomes are present in its cell. So, when meiosis is seen to produce the sperm cell, the number of chromosomes will become half. That is, it will be 48 by 2 and that will be 24. Therefore, children, the number of chromosomes in each sperm cell will be 24 and our correct answer is thus A, that is 24. Okay, let me quickly revise this one more time. Children, sperms are produced by meiosis and not mitosis. Very important thing that you need to keep in mind. Sperms are produced by meiosis. Number 2, meiosis is a reductional cell division. That is, the number of chromosomes become half. Point number 3, in gorilla, the number that we have been given over here is that there are 48 chromosomes. Therefore, after meiosis, the number of chromosomes that will be found in the sperm cells will be 48 divided by 2. That will be 24. I hope children, you could follow this explanation. Okay, therefore the correct answer will remain A, that is 24. Okay, now let's look at the next question. Pay attention, now this is a 3-marker question asked in the year 2024. So let's solve it. The diagram below represents a stage in mitosis. We are talking about mitosis over here. Identify the stage given above. Give one reason to support your answer in A and mention the number of chromosomes which are given in the diagram. Okay, children, so let's solve this question. Children, so now let's look at the solution. Now, we will quickly draw the diagram of how a cell looks during its division, starting from the prophase, which will help us come to our answer very quickly. So, now let's take this as a normal cell. Okay, this was a cell who has decided to undergo cell division. We know that it has a nucleus and in the nucleus, it has the DNA. Now, children, during the prophase, what will happen? The DNA condensation is seen. It becomes short and thick and it forms the chromosomes. Yes, so we will get to see something like this. Also, the nuclear membrane disappears, so we will not be able to see nucleus anymore. The condensation is a long process and we also get to see at the end of prophase its sister chromatids. So, this is how a prophase would look like. Now, during the metaphase, children, what do we get to see? We get to see the centrioles, which will come at the poles, right? It will give rise to this spindle fiber. Some are continuous and some are discontinuous. So, these are the continuous ones who moves from one pole to another pole. In the center, let's draw this discontinuous one. So, here are these chromosomes which are attached with the discontinuous fibers, and as we can see, the spindle fibers, they bring the chromosomes towards the equatorial plate, right? So, we cannot see this diagram over here. That means this is neither prophase, this is neither metaphase. Now, children, after metaphase, the next phase we get to see is the anaphase. Okay, now let's see what happens in the anaphase. Children, we know that the chromosomes are attached to each other at a constriction which is known as a centromere. Alright, so let me draw the chromosome over here, and this is the point where it is attached. And on this centromere, we have the spindle fiber which pulls, and as a result, it separates or it cracks the centromere into two halves. Okay, so let's see over here in a main diagram. So here is our centrosome. These are the continuous fibers of the centrosome, and these are the discontinuous fibers of the centrosome. Now what has happened, since the discontinuous fibers are pulling at the centromere, it cracks the centromere. So this centromere divides into two parts, which we can see over here, and each centromere is carrying half of the chromosomes with it. And this is exactly what our diagram looks like, right? And therefore the phase that we are talking about over here is the anaphase. So we can see over here, this over here, these are the centrosomes, these are the continuous fibers, and these are the discontinuous fibers which are attached to the centromere, right? Now, since there was a pull by the contraction of the discontinuous fibers, the centromere divided into two halves. And with each half, one chromosome went on the upper side and another chromosome went on the lower side. That means the chromosome, as we can see over here, children, it is broken into two parts and they are being pulled in opposite directions. That's exactly what our diagram is over here. Okay, so this phase is the anaphase. I hope the explanation of it is also clear, where the discontinuous fibers have pulled and separated the chromosomes from each other. Okay, so here was the contraction of discontinuous fibers, thus the chromosomes are separated and pulled at the respective poles. Alright, now the question 3 was, total how many chromosomes are we seeing in this main diagram asked in the question. So we can, let's count quickly. This is chromosome 1, chromosome 2, chromosome 3, chromosome 4, 5, 6, 7 and 8. So total how many chromosomes we can see? Total we can see 8 chromosomes. I hope children this explanation was crystal clear to you. Yes? Okay. Chalo, then let's move ahead. Now we have a 1 marker question which was asked in the year 2025 in the form of a MCQ. Select the correct answers to the questions from the given options. A phase of cell division where the spindle fiber disappears is option A telophase, option B prophase, option C metaphase and option D is anaphase. Children, are you able to answer this question? If you paid attention in the last question, you would be able to answer this question. Yes, the correct answer is telophase. Okay, so children, let's understand quickly why telophase is the right answer. So children, what happens in a prophase? Prophase is where the DNA condensation takes place, where the chromatin or thin threads of DNA, they condense to form short and thick chromosomes which are attached at the centromere. This is a prophase. Now what happens in the metaphase, children? The spindle fiber starts to appear, right? So for that we need the centrosomes at the two poles and the chromosomes, we can see over here, they are attached to this spindle fibers. So spindle fibers are seen in the metaphase. How telophase takes place? So what happens in the telophase? The spindle fibers contract and they pull the chromosome upwards, right? So this is this chromosome moving towards this centriole and this is the another chromosomes which are moving towards this centriole. So here also we can see the spindle fibers. And the last phase that we have is the telophase. Telophase is essentially reverse prophase, meaning whatever is happening in the prophase, its reverse is taking place in the telophase. That is, the chromosomes, they will start to decondense and they will convert again into this chromatin fibers of DNA. So, we will be able to see the chromatin fibers. The nuclear membrane will start to appear and so will all the other cell organelles. Okay, and therefore, the correct answer is the telophase, where these spindle fibers disappears. I hope this explanation was clear. Alright children, so now let's move ahead. Now let's quickly solve again a 1 marker question asked in the year 2025. The question is, read the explanations given below and name the structures. Example, the tubule which produce the sperms. The answer is seminiferous tubules, correct. The point of attachment of two chromatids of a chromosome is, so the question over here is, what is the point at which the two chromatids are attached? So we know that children, this is a duplicated chromosomes, or we can take a very simple chromosome which has not yet duplicated. So the point where these two chromatids, they are attached. We have already solved this question which was asked in a different format, but the answer still remains the same, that is the centromere. Okay, centromere is a point where the two chromatids of the chromosomes attach with each other. Are we clear? Okay, chalo, let's move to the next question. Now we will solve a 2 marker question which was asked in the year again in 2025. Let's look at it. Mention the 2 pairs of nitrogenous bases that are present in a DNA strand and they pair with each other by the hydrogen bonds. Okay, for that we need to know the nitrogenous bases which are found in the DNA. Can you recollect any? Yes? Alright, so let's quickly mention. We find adenine, we find guanine, we find cytosine, and we find thymine. These are the 4 nitrogenous bases that we find in the DNA. Please remember, in the DNA and in RNA, there is a slight difference. Okay, instead of thymine, in RNA we find uracil. Okay, anyways, the question asked of here is that which are the 2 pairs of nitrogenous bases found in the DNA and how they are paired with how many hydrogen bonds. So please remember children, adenine always pairs with thymine. It loves thymine like anything. Okay, adenine loves thymine. Alright, and guanine, it loves cytosine. So they have already selected their partners. Adenine goes with the thymine, guanine goes with the cytosine, right? But there is a slight difference. That is, the adenine and thymine, they don't have a very strong bond. Therefore, adenine and thymine, they are attached to each other just by 2 hydrogen bonds. But guanine's love for cytosine is very high and therefore, guanine is attached with the cytosine with 3 hydrogen bonds. Okay, so guanine and cytosine are attached with 3 hydrogen bonds because their love is very strong, and adenine and thymine, they relatively have a weak bond, therefore they only have 2 hydrogen bonds. Okay, they have weak love towards each other. Alright children, let's move ahead. Now let's solve a 3 marker question which was asked in the year 2025. Draw a neat labeled diagram of an animal cell showing metaphase with total 4 chromosomes. Alright, we have already drawn this a couple of times. Let's quickly draw it again. Children, here we can see the diagram of a cell during the metaphase. At the top and the bottom fold children, we can see the centrosome. Alright, so here also this is the second pair of the centrosome or the centriole. From them, we are getting the astral rays which are forming this spindle fibers. So we can see over here the spindle fibers coming from the top and it is attaching with the centromere of this chromosome, and similarly we are having this another spindle fiber coming at the bottom and attaching to the same centromere. So all the spindle fiber children, they are attaching with the centromere of the chromosomes. Alright. Second thing, we can see that all the chromosomes, they are lying at this equatorial plate or the equatorial plane. Right children? Over here, this is the cell membrane, and over here we are seeing all this will be the cytoplasm throughout the cell. So this is what a cell will look like in the metaphase. Keep one thing in mind, in the metaphase, the first thing that should be imagined in your head is that the chromosomes are in the equatorial plate. Any diagram you see with that is simply your metaphase. Alright children, I hope this is clear. Let's move ahead. What is the structure of DNA as proposed by Watson and Crick? So children, Watson and Crick, they gave us the double helical structure of DNA. Where two DNA strands, they helically wound around each other. Next question, again a 1 marker question. Define the term allele. Now lots of students makes a difference, and even today I can bet half of the students don't understand the difference between gene and allele. So let's quickly understand it. Children, gene is a segment of DNA. Please remember, it's a structure. It's a segment of DNA which is present on a chromosome. So let's say this is a chromosome. Alright, and this particular is a segment of chromosome which represents a gene. This is, for example, let's say gene for eye color. Okay, now this is a gene for eye color. A gene is a segment of DNA, children. That gives rise to a particular characteristic of a body or a particular character of a body. Let's, for example, we've taken the character as the eye color. So we will all have a gene which will help in developing that eye color, whether it will be blue or it will be brown or it will be black or grayish or different shades of brown. Yes, that's a gene. Then what's our allele? Children, allele is a variant of gene or it's an alternative of a gene. Meaning, let's say you have allele which gives rise to brown eye color. Okay, then your eyes will be brown in color. Let's say you have an allele that gives rise to green eye color. Then your eyes will be green in color. So alleles, they are variants of gene. But what's a gene? It's an eye color. It's a trait. It's a character that is going to be developed in your body. Alright. So gene is having a information which will develop a particular character in your body. But how will it develop? What it will look like? That will be decided by the allele. So if you have allele for brown eye color, your eyes will be brown. If you have allele for blue eye color, your eyes will be blue. So every gene comes with its allele. So what is gene, children? Gene is a segment of DNA that will give rise to a particular character of your body, which for example is the eye color. And allele will decide how that character will look like, whether it will be blue in color or whether it will be green in color or whether it will be brown in color. Okay. So in simple words, gene is a structural unit of DNA and allele is a functional unit of DNA. It is essentially the same thing. Alright. But one will decide the trait, one will decide how the trait will appear like. So it is same, but it is still different in its own way. I hope this was clear. Alright children. So now let's move ahead. Now let's start with a third topic, that is genetics, the basic fundamentals. Let's look at it weightage. So in the last 2 years, children, you can see this is a very high weightage topic. Total 4 questions of 1 marker each were asked in the year 2023. 2 questions of 2 marker each. So total 8 marks were asked from this topic in 23. In 24, 2 questions of 1 mark each and 1 question of 3 mark was asked. Total 5 marks was asked from this topic. And in 2025, 2 questions of 1 mark each and 1 question of 3 marks. So again, total 5 marks from this topic have been asked. Let's look at it in a graphical manner to make it even more simpler. So we can see over here, total 4 questions of 1 mark and 2 questions of 2 marks were asked in 23. In 24, 2 questions of 1 mark each and 1 question of 3 mark was asked in year 24. And in 2025, 2 questions of 1 mark and 1 question of 3 mark was asked. Alright children, now that we've understood the weightage of this topic, let's solve their PYQs. Now in the year 2023, MCQ was asked for 1 mark. Let's look at the MCQ. The sex chromosome in a human ovum, that is the gamete they are asking, is X chromosome, Y chromosome, both X and Y chromosome, either X or either Y chromosomes. The female gamete, that is the ovum, has X chromosome and the male gamete has either X or it has Y chromosome, one of the either. Why is that so? Because when you look into the female, children, when you look at the sex chromosome of the female in any cell, not just the ovum, in any other cell, it will always be XX. And when you look at the sex chromosomes of male, any cell other than the sperm will always be XY. Okay, so females have XX chromosome in their cell, males have XY chromosome in the cells. Now, when this cell undergoes meiosis to produce the ovum in case of female, only 1 chromosome will enter into the gamete, so it only has to be X, right? And when the male cells undergoes meiosis to produce the sperm, it will have either of this gamete, so it will either have X chromosome in the sperm or either the sperm will have a Y chromosome. Okay, now since we are asked about the female gamete will have the X chromosome. So the correct answer over here is option A, right? But if they are asked for the male chromosomes that which will be found in the sperm, then option will be D, that is either X or either Y chromosomes. I hope children this is clear. Alright. Okay. So the correct answer is A. Now let's look at the another MCQ. Haploid number of chromosomes are found in. So the keywords over here is haploid number. Haploid means single N. That is 1 set of chromosomes. So let's look into it. Option A, nephrons. Option B, neurons. Option C, skin cells. And option D, sperms. Children, all the cells of our body are diploid. All the cells of our body are diploid other than the gamete cells. Over here, the only answer that is mentioning a gamete cell is option D. So, sperm is a gamete, also known as the sex cell. They are produced by meiosis, which is the reductional division, right? They are produced by meiosis, which is a reductional division. And therefore, they will only have 1 set of chromosome, that is haploid number of chromosomes. They will show haploidy nature. Alright, so the correct answer over here is D. Nephrons, neurons and skin cells, they do not produce gametes. Therefore, they are diploid. They will have 2n number of chromosomes. Alright, but sperms or ovum, these are gametes. They are produced by meiosis or reductional division. Therefore, they will only have one set of chromosome, that is, they will show haploid number of chromosomes. Right? So, the correct answer is D over here. Now, let's look at the next MCQ. Arrange and rewrite the terms in each group in correct order to be in a logical sequence beginning with the term that is underlined. Xylem, soil water, cortical cells, root hair. Alright children. So, let's quickly arrange them. So, first we will have the soil water that will be absorbed by the root hair. The water will move into the cortical cells, and then from the cortical cells it will move into the xylem. Pretty straightforward question, I guess children. Yes. So, let's move to the next question. Now, we have a 2-marker question asked in the same year. State Mendel's law of segregation. Alright, let's answer it. Children, according to the law of segregation, only one allele will enter into the gamete cell during meiosis. Right? Now, let me explain this statement with an example. Let's say a particular seed is round and it is yellow in color. Right? So, during gamete formation, only one of the 2 alleles will enter. That is, from here only one allele, that is R, and from here only one allele, that is small y, will enter. This is your law of segregation. Let's move ahead. Alright children, so now let's look at the next question which was asked for 2 marks. The question is, state Mendel's law of segregation. Please remember children, this is the Mendel's first law. Alright. This is the Mendel's first law. Now, what does law of segregation states? Law of segregation states that the 2 pairs of alleles separates from each other during gamete formation. And only one of it enters into the gamete. So, let's understand this law with an example. Let's say a particular seed is round and yellow in color. So, this is how the genotype would look like. Right? Now, when meiosis will take place, children, only one of the 2 alleles will enter. Now, let's understand this. Children, RR, they are the alleles for round shape. Always remember, a gene has 2 pairs of alleles, right? And similarly, these are the alleles for yellow color. Now, when meiosis will take place, children, only one of the 2 alleles will enter into the gamete. That is, from here, these 2 alleles will separate, and similarly, these 2 alleles will also separate, and only one of the 2 alleles will enter into the gamete. So, let's say this is a sperm. So, only one of the 2 allele will enter and only one of these 2 allele will enter. Or if ovum is being formed, then similarly we will have the same scenario. So, these 2 alleles, they are separated from each other and only one of the 2 alleles will enter into the gamete. And this is the law of segregation. Therefore, the gamete will have one allele for the shape and one allele for the color of the seed. Next thing to remember, children, This segregation or this separation is seen during the anaphase of the cell division. Please keep in mind. Now, you might think that why is this necessary? Because children, a gene can only have two alleles. Now, let me explain that. Let's say this is the sperm and let's say this is the ovum. During sexual reproduction, the sperm and ovum, they will fuse with each other. And now, this will also come together. So, we will again get the same condition, or we will get the same gene, each gene having two alleles. So, for example, the gene for shape will now be RNR and gene for color will be Y and Y. So, this gene of shape is now having two alleles. This gene of color will now have two alleles. So, that restores the same condition. Children, if there was no segregation, what would happen? The sperm would have RR and YY and the ovum would also have RR and YY. And now when they fuse, look at the condition we would get RR, RR, Y, 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 Y. This is not possible because one gene cannot have four alleles in a healthy organism. Okay, so this is absolutely wrong. Therefore, the alleles must segregate during gamete formation, which however during fusion of male and female gametes will come together and restore the condition. The condition being that each gene will have two alleles. I hope this is clear. Alright, now let's move to the next question. Define the term mutation. So let's quickly answer. Children, mutation is a sudden, heritable and a permanent change seen in the genetic material. Or let's say the DNA. Okay, so there's a sudden, inheritable and a permanent change. Now, what am I trying to exactly mean by that? Children, let's look at this, a strand of DNA. This is the 5' end, this is the 3' end. Okay, and it has the following nucleotides with the nitrogenous base A, G, T, C, 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 A and T. Now, due to presence of certain mutagens, mutagens are chemicals that can cause mutation. Now, this chemical could be excessive exposure to UV rays or excessive exposure to X-rays, or it simply could be tobacco, okay, MSG salt. So, all these are mutagens which are seen to produce changes in this DNA. Like for example, it may add a nucleotide. So, here suddenly a new nucleotide has been added. This is also mutation, alright, or it may delete some nucleotide. So, let's say this was your original sequence of nucleotides and now this 2 sequence is deleted from here. So, addition of a nucleotide, deletion of a nucleotide or exchange of nucleotide. That is, let's say for example children, this was a healthy nucleotide, that is A, G, T, C. Now, instead of 2 C's, we have T's. So, this is replacement or exchange. So, whenever the normal sequence of a DNA undergoes some changes, that is either addition of new nucleotide in this case or deletion of nucleotides in this case or replacement or exchange of nucleotides, then this all leads to mutation. Now, this mutation has occurred because of excessive exposure to UV rays, X-rays, tobacco, MSG, etc. There are another hundreds of mutations which are present and we are surrounded by it. right? So when we are exposed through these mutations, we are at a risk of developing such changes in the nucleotides, which are sudden, heritable, meaning we can pass it to the next generation and they are permanent. And these changes are known as mutation. Okay, children, I hope this is clear. Now, let's move ahead. So now let's look at the 2-marker question which is asked in the year 2023. A pure breeding red flower variety of the pea plant, capital R, is crossed with a pure breeding white flower variety of the pea plant, that is small r, small r. Draw a Punnett square to find out the phenotypic and the genotypic ratio of the progeny belonging to the F2 generation. Alright, so let's quickly do it. We have two parents, one with the genotypic ratio capital R, and another parent with the genotypic ratio small r, okay. This is the red flower parent and this is the white flower. This is the red flower parent and this is the white flower parent. Now, when we cross them, we will get the progeny who will have the genotypic ratio big R and small r, okay. This is going to be the F1 generation. Now, in order to get the F2 generation, children, let's quickly draw a Punnett square to make this very simple. Let's say this is the female gametes over here with the alleles this, and these are the male gametes over here with the same alleles. Now, when we cross it, what will we get, children? We will get big R, big R, small r, big R, small r and small r, right. So, these are the F2 generations who will look like this. Now, we are supposed to find out the genotypic ratio of the F2 generation and the phenotypic ratio of the F2 generation. So, let's quickly look into it. Now, what are the genotypes we are getting, children? We are getting one capital R, then we are getting two big R, small r and one small r. So, genotypic ratio will be 1:2:1. Alright. Now, let's look at the phenotypic ratio, children. Here we are getting one capital R and two capital R and small r. All this will bear us red color flowers. So, that will be 3 and only one will bear us white color flower, that is small r. So, the phenotypic ratio will be 3:1. I hope this was clear. So, let's again look at this solution. We will get the same answer. So, the phenotypic ratio in the F2 children is 3:1 and the genotypic ratio in the F2 generation will be 1:2:1. So, over here we can see there is a red flower and the alleles for it is double R capital, and here we have the white flower and the allele for it is small r, right. When you self this flowers, we will get the F1 generation where all the flowers will be red flower and the genotype will be big R, small r, right. Now, when we will do selfing or when we will do self pollination of this flower, we will get, as we can see over here, 3 red color flowers and 1 white color flowers, and therefore the phenotypic ratio is 3:1 where 3 are red color flowers and 1 is white color flowers, and the genotypic ratio will be 1:2:1. This is the double R capital, this is one capital R small r and this is small r. I hope this was very easy peasy for you. Yes. So, now let's move to the next question. Now, this is a MCQ asked for 1 mark in the year 2024. A zygote which has Y chromosome inherited from the father will develop into, option A, will depend on the chromosome inherited from the mother. Option B, will develop into a girl. Option C, it may develop either into a boy or a girl, and option D, it will develop into a boy. So, let's quickly solve it. Children, the mothers have chromosome XX, the father have the sex chromosome as XY. The mother can only and always will give X, father can give either X or father can give either Y. Alright. Now, since father is giving Y in our situation and the mother is always going to give X, so here the zygote will most definitely, in fact it will be Alright, therefore the correct answer is D. I hope this was clear, children. Okay, let's move ahead. Now let's look at the another MCQ. Four friends P, Q, R, S were discussing the examples of the genetic disorder. The examples they quoted were as follows. P stands for color blindness and malaria. Q stands for albinism and cholera. R stands for hemophilia and color blindness, and S stands for hemophilia and albinism. Who gave the correct example? Alright, children. So P gave the examples that color blindness and malaria, they are genetic disorders. R gave the example that albinism and cholera are genetic disorders. R said you both are wrong. According to me, hemophilia and color blindness are the genetic disorder. And S said no, I think hemophilia and albinism are the correct genetic disorders. So we have to find out who gave the correct answer. So let's look at the option. According to option A, P and Q are correct. According to option B, R and S are correct. According to option C, P and R are correct. And according to option D, Q and S are correct. So let's quickly look into it. So the friend with name P says color blindness and malaria are the correct genetic disorders. I agree with color blindness, and I'm sure even you, but malaria is not a genetic disorder. It is an infectious disorder. It's a communicable disorder which is caused by plasmodium carried by a mosquito. Alright. Friend Q says albinism and cholera are the correct genetic materials. I agree with albinism, but I do not agree with cholera because cholera again is a bacterial disorder which is communicable in nature, caused by the bacteria Vibrio cholera. So again, this is wrong. Friend R says that hemophilia and color blindness are genetic disorders. I agree with it. And S says that hemophilia and albinism are genetic disorders. And therefore, both R and S are correct, and the correct answer is option B, that is R and S. Let me tell you a little bit detail about hemophilia and color blindness. Children, hemophilia and color blindness, both of them, they are X-linked recessive disorders. Meaning, on the X chromosomes, children, we will find their recessive genes, okay, which leads to these diseases, that is hemophilia and color blindness. Children, albinism is because of the mutation found in the autosomes, which are not the sex chromosomes, but other than the sex chromosomes, the other 44 chromosomes that we have, those are known as autosomes. So albinism is an autosomal disorder. Therefore, children, friends who are R and S, these two children, they are the smartest among their friends, and therefore option B is the correct answer. Alright, students. Now, let's look at 3-marker question asked in the year 2024. Given below is a schematic representation of the inheritance of the shape of seeds. Pay attention to this, of garden pea, and we have to answer the following questions. So, we can see over here, two parents were used. One parent has a round shaped seed and another has a wrinkle shaped seed, right? So, let's put it like this. We'll put capital R for round shape and small r for wrinkled. Alright. Then we get a F1 generation, and then on self-pollinating the F1 generation, we will get F2 generations. So now let's look at the questions that we are supposed to answer. Question A is, which is the dominant and the recessive allele of the trait? Next, what does the ratio 3:1 in F2 generation represent? Third, state the Mendel's law of dominance. Alright, chalo. So let's answer this question. Children, we can see over here, as this is the round seed And this is a wrinkle seed. Again, we have used the same alleles, capital R for round seed and small r for wrinkle seed, right? Then during reproduction and gamete formation, the alleles will separate from each other. So we can see over here separated alleles on both the sides. Alright, and on fusion of gametes, we will get the F1 generation. Each of it will have capital R, small r. On self-pollinating the F1 generation, we will get 4 types of F2 generation. That is one with capital R, capital R, capital R, small r, capital R, small r and small r, small r. If you are not able to follow this, let me solve this quickly for you so that it becomes very easy for you to understand. So we have a round seed and we have wrinkle seeds. Alright. On gamete formation, we will only get one of the alleles which will come together, and therefore in the F1 generation, all of the seeds will have round shape. This is the F1 generation. To get the F2 generation from the F1, let's quickly make a Punnett square to make this simple for you. Okay. F2 generation is obtained by self-pollination, and over here also the alleles will separate. Okay. So here we may have in the male parent, we will have 2 separated alleles, and in the female parent again we will have 2 separated alleles. So let's look at the 4 types of plants that we may get. We may get capital R that we can see over here. Then we get capital R smaller that we can see over here. Then again capital R smaller and small r, small r. So we got these 4 types. Now the question they had asked was, why is the F2 phenotypic generation is 3:1? So let's quickly understand, children. Over here, first of all, the F2 generation, the progeny with double R will have a round seed. The second progeny, that is big R, small r, will also have a round seed, and similarly the third progeny will also have a round seed, and the fourth progeny is having small r will have a wrinkled seed. So here the 3 stands for 3 progeny with round seeds and 1 stands for 1 progeny with the wrinkled seed. Now we have understood this, right? We have understood this particular part. But how is that big R is round and why small r is wrinkled? For that we need to understand which allele is dominant, which allele is recessive. Children, in allele there is always a fight, right? So whenever you see that the alleles are written in capital, that means those are the dominant alleles. और जो allele dominant होता है children, वो हमेशा उसकी चलाएगा, meaning the dominant alleles are always expressed. So if the dominant allele is for the trait of roundness of a seed, then the seed will be round. Okay. Such a condition, children, double R which are both capital, this is known as homozygous dominant. Now let's look at another condition, that is big R and small r. Now children, big R, like I told you, it represents the dominant allele, right? Whereas the small r, it represents the recessive allele. Recessive allele meaning weak allele. So when a weak person is fighting with a strong person, who will win? The strong person, right? So that's exactly what's happening. When we have 2 contrast alleles, that is we have a dominant allele and a recessive allele, the recessive allele cannot take over. That is, the dominant allele will express itself, and in such a case also the plant will show round seed. Such a condition is known as heterozygous dominant. Hetero means different, right? So here we have, this is the dominant allele and this is the recessive allele. Now if we look at such a condition where we have 2 small r's, children, over here both the alleles are weak, both the alleles are recessive, there is nobody to dominate. So only in this condition the recessive allele can express itself, meaning in this case the seed will be wrinkled and it will not be round and smooth. Alright, this condition is known as homozygous recessive. Alright children. So, who is the dominant allele in our case? R is the dominant allele and the small r is the recessive allele. I hope this is clear. So, we have answered our first question, that is the big R or the capital R is the dominant allele and the small r is the recessive allele. Now children, in the second question, it was asked that why do we get 3:1 phenotypic ratio and what does it mean? So, over here we have already solved that on selfing the F1 progeny. On selfing it children, we get the F2 generation where we are getting 3 progeny who will have round seeds and 1 progeny who will have wrinkle seed, and therefore we get the phenotypic ratio as 3:1. Okay, all right. The last question was state the law of dominance. Children, what does the law of dominance says? Mendel said that whenever there are contrasting allele of one gene, that is when one allele is dominant and one allele is recessive, only the dominant allele will be able to express itself, that is express that particular characteristics, and the recessive allele will be inactive. Alright, its expression will be masked, it will be hidden by the dominant allele. So, what is the law of dominance? That whenever we have contrasting alleles, as we can say over here in this case, here we have a contrasting alleles where one is a dominant allele and one is a recessive allele. In such situation, only and only the dominant allele will express itself and therefore we will get the round shape seed in this particular case. Alright students, I hope this was clear. Now, let's move on to the next question. Let's look into it. A scientist wanted to conduct a genetic experiment on mice. He cut the tails of all the mice and made them reproduce. What will be the phenotype of the offsprings? Phenotype meaning how will the mice look like? Will they still have a tail or they will be born without the tails? So, let's look at the option. Option A, all the offsprings will be tailless. Option B, there will be a mix of tailed and tailless offsprings. Option C, all the offsprings will have tails. And option D, tail of the offsprings cannot be predicted. So, let's solve this question. Children, cutting of the tail after the mice is born, this is an acquired character. That means the mice was born with a fully developed tail. Okay, you have cut it after it was born and therefore such a character is an acquired character. And any acquired characters or any characters that we develop over a lifetime, we cannot pass it to the next generation because these characters are developed not due to some genetic element. It is not because of presence of a particular gene. So, when there is no change in the gene giving rise to that new character cannot be passed on to the next generation. Like for example, let's take Olympians, right? The great Olympians. Now, these Olympians, they have developed a amazing physique and muscle mass to be expert at their sports. Now, this is a change. This is a development that they have made over a lifetime. They were not born like that. They have developed, they have worked hard, they have practiced and made themselves great. Does that mean that this Olympian will be able to pass all of this to the next generation? No, because building muscle mass was something he did throughout his life. He was not born with that great physique. He had to build it. Similarly, the mice was born with a tail. So, during his lifetime, if we cut the tail and make it reproduce, the offsprings will still have a tail because the genes of tail is still present as it was in the parent, it was inherited as it is in the offspring. So, if there is no change in the DNA of the sex cells, that is the sperm and the ovum, there will be no such inheritable changes seen in the offsprings of those parents. Alright. Therefore, children, all the offsprings of this mice will still have tails because acquired characteristics, they are not passed on to the next generations. Alright. Therefore, the correct answer here will be option C. Okay. Let's move on to the next question. Now we have another MCQ. Ajay and Vijay are siblings. Ajay can roll his tongues while Vijay cannot. What is the genotype of their parents? Option A, both the parents are homozygous dominant. Option B, both the parents are homozygous recessive. Option C, one parent is homozygous dominant whereas the other is homozygous recessive. And option D, both the parents are heterozygous dominant. Now, this question will require some calculation. Let's get into it. Children, rolling of the tongue is only possible because of a presence of a dominant allele. So, let's say if Ajay can roll his tongues, meaning he might have a genotype which capital R, capital R, capital R, small r. In both this case, he will be able to roll his tongue, right? In case of Vijay, he cannot roll his tongue, meaning his genotype will be smaller. Because it is recessive, he will not be able to roll his tongue. Now, let's see in which of the following combination of the genotypes of the parents such a genotype of children can be obtained. So, let's start from option A. The option A says both the parents are homozygous dominant. That means the mother is also capital R and the father is also double R, capital R. They are homozygous dominant. Now, children, in this case, what will happen? Both the gametes will consist of capital R and all the children will have the genotype capital R. That means Ajay will also have capital R and Vijay will also have this condition, which according to the question is not there. Therefore, option A is not the correct option. Now, let's look at option B. According to option B, both the parents are homozygous recessive, meaning father is also smaller, mother is also smaller. Therefore, the zygote that will be formed in F1 generation will also be smaller, which is not the case because Ajay is bigger and Vijay is smaller. So, in this case, this is also not right because if this was right, how could Ajay be showing this genotype? That is not possible. Therefore, option B is also not possible. Option C says that one parent is homozygous dominant and the other is homozygous recessive, meaning one parent is homozygous dominant and one parent is homozygous recessive. In this case, the zygote that will be formed will show this genotype, that is bigger and smaller. That means both Ajay and Vijay would be able to roll their tongues. You can see over here, they would be able to roll their tongues. Again, that's not what the question says. So, by default, option D is the right answer. But we will also solve it to be fully sure. So, let's look over here. Both the parents are heterozygous dominant. That means we will have this condition. This is a heterozygous dominant condition. Yes or no? So, in this case, what we may get? We may get this condition, this genotype. We may also get this genotype. We may also get this genotype and we may also get this genotype. So, now let's see whether any of this genotype matches with Ajay and Vijay. Ajay is either homozygous dominant or heterozygous dominant. Yes, we can see over here. Let's check for Vijay. Vijay is homozygous recessive. Yes. Therefore, answer D is the right answer. Got it, children? So, option D is our correct answer. I hope this was clear. Now, let's move on to the next question. Now, this is a 3-marker question. Pay attention. A farmer cultivates a unique species of tomato plants. The two traits selected for breeding were the color of the fruits and the height of the plant. The farmer initially crossed a homozygous tall plant bearing red fruits, that is double T double R, with a dwarf plant bearing yellow fruits, that is a small t small r. And the genotype of the offsprings of the F1 was big T small t and big R small r. Next, he crosses two F1 generations Explain the phenotype of the offsprings of the F1 generation obtained from the initial cross. And question B, mention the phenotypic ratio of the F2 generation offsprings. Alright. And option C is state Mendel's law of independent assortment. Alright. So huge questions, lot of calculations. Let's do it fast. Okay, children. So the farmer crossed tall plant with red fruit with a small plant or the dwarf plant with the yellow fruits, right? And the F1 generation he got was heterozygous condition, and therefore this particular plant will be tall and show red fruits. Now, how is that possible? Children, remember we talked about earlier that every gene has two alleles. One allele will be dominant or one allele will be recessive, or both of them can be dominant or both of them can be recessive. In this case, children, we are getting one dominant allele and one recessive allele for the height of the plant. Always the dominant allele wins the fight. Anybody who is dominant will win the fight and things will go as per them, right? So, when we talk about the gene of the height of the plant, the dominant allele is the tall allele. So, even when there is a recessive allele present, it will not be expressed. Only dominant allele will be expressed and therefore the plant will be tall. Similarly, for the color of the fruit, we have two alleles for it. We have a dominant allele and we have a recessive allele. The dominant allele stands for red color and the recessive allele stands for yellow color. So, whenever we have two contrasting alleles as per the law of dominance given by Mendel, it will be the dominant allele who will express itself. Therefore, the progeny of the F1 generation will be tall and show red fruits. Alright. So, I hope this is clear. So, here we will get the answer, tall plants with red fruit. Now, let us look at the second question. Mention the phenotypic ratio of the F2 generation offsprings. So, let us quickly make a Punnett square. Okay, so this will be the male gametes. We will get the following combination. Same combination we will get for the female gamete. I would urge you students to make this Punnett square along with me so that you understand this even more better. Alright, now let us look at the different phenotypes that we will find over here. Let us look at the first one. Now this is double T double R. This is homozygous dominant for tall and red fruits. Okay, so let's see how many other phenotypes we may get identical to this. This will also be tall and red fruits, that is number 2. This will be tall and red fruits. Similarly, tall and red fruits, tall and red fruits. So total 5 we've got so far. Tall and red fruits, tall and red fruits. So this is total 7. Tall and red fruits 8. Tall and red fruits 9. So total we've got 9 tall with red fruits. Alright, so these are tall plants with red fruits. Now let's look at the next phenotype. This will be tall with yellow fruits. Similarly, tall with yellow fruits. Alright, and this will also be tall with yellow fruits. So these are tall plants with yellow fruits. Total we will have 3. Now let's look at the next phenotype. Yes, this will be dwarf with red fruits. This will also be dwarf with red fruits, and this will also be dwarf with red fruits. So total we have 3. And we will only have one dwarf with yellow fruits. Therefore, what will be our F2 phenotypic ratio? It will be 9:3:3:1, where 9 stands for tall plants with red fruits, 3 stands for tall plants with yellow fruits and dwarf plant with red fruits, and 1 stands for dwarf plant with yellow fruits. I hope this was clear and fun. Now let's look at the last question. State Mendel's law of independent assortment. Now let's quickly solve it. Children, Mendel's law of independent assortment, it tells us that when more than one traits has to be inherited, the alleles of each trait or the alleles of each gene will separate independently and is not dependent, or it is not affected by the separation of the other alleles of another genes. Let's take for example, children, over here we saw we had two plants, right, where we had taken two traits. We had taken the height of the plant and we had taken the color of the fruit. Yes or no? So over here there are two traits which are to be inherited, right? During the anaphase or during gamete formation, children, what happens? These alleles are separated. That means allele of this trait will also separate and allele of the second trait will also separate. So the separation of each of this allele is independent of each other. That is exactly what the Mendel's law of independent assortment teaches us. I hope this was clear, children. Alright. Yes. So we can see over here, children, this is the same diagram. We have done the working. Here in the left we can see the tall plants with the red fruits, and on the right we can see the dwarf plant with the yellow fruits. So children, we have done the working of the F2 generation already, and I hope it is clear. Okay children, so with this we have finished our PYQs on the above topics, and I will see you soon. Till then, take care and stay curious.